Hello, everybody. Katia Plant Scientist has a big update for you. I am now not only just Katia Plant Scientist, I am Dr. Katia Hougaard, the plant scientist. Yes, at long last, I have finally completed my PhD. I wanted to share this joyful news with all of my fellow plant enthusiasts who have so kindly followed, supported, liked, and commented my science communication content here on social media. I just want to let you know that although I'm now Katia Plant Scientist with a PhD attached at the end, I'm still going to be bringing you lots of exciting plant science communication content for years to come. I would like to back up a little bit and talk about what it means to do a PhD. PhD stands for Doctor of Philosophy. A PhD project is a multi-year educational project at the postgraduate level that requires students to take a research question, a problem in science, something that is unknown, and devote years of study and research into finding out as much as they can about it. I'd like to tell you a little bit about the research that went into my PhD. My PhD project focused on the plant immune system, and my PhD's main questions revolved around what are known as resistance genes, or R genes, in plants, which give them resistance to specific pests and pathogens. My model system was the plant Medicago truncatula, a relative of clover and peas and beans, and its innate resistance to the pea aphid, which is an economically important pest of many legume crops. I did research in the lab using molecular biology to study the genes themselves and also to do experiments with plants and aphids. For example, finding out how the plant's resistance changed when they were subjected to different kinds of stress. I also used a lot of bioinformatics in my works. And I use the bioinformatic techniques to find out possible ideas for how the defense response might work, like what chemicals are involved and what biological processes lead to plant resistance to aphids. I could not do a PhD alone. In order to do a PhD, a student must pair up with a supervisor. The supervisor is a research mentor. It's more than just simply a college professor. This person is the mentor who teaches you how to do science, provides helpful feedback on your work, and they teach you with the aim of helping you become an independent researcher yourself. I'm very grateful to my PhD supervisor, Professor Colin Turnbull at Imperial College London, for his supervision, guidance, mentorship, and friendship during the very long journey we've both been on. Now, I have sometimes mentioned in some of my updates for my social media that I had to go on hiatuses in order to focus on my work. By the time I started making social media content, I was already kind of transitioning from the lab-based research phase into the writing up phase. But around that time, some really difficult things happened in my personal life that meant that I had to go on some interruptions of studies too. So it's been a longer than planned journey to complete this PhD. I started out by doing research based in the lab using hands-on molecular biology techniques, as well as working with the plants and aphids. When I had gathered enough data, I could start writing the thesis. And let me now tell you a little bit about how a thesis is structured. I have the thesis. This is at least the first draft of it. I'm going to do a bit more editing before it's made official, but let me show you the title page. Of course, there's a lot more to a thesis than a title page. Inside a thesis, there is an introduction section that introduces the type of research we are doing and the field of science that I'm working in. Next up, we have a general methods and materials. The general methods and materials talks about some of the lab techniques and bioinformatics techniques that I used. And next, we have several chapters where we tackle the different research questions. So in this case, we talk about the identity of the resistance genes. Then I talk about transgenic plants that I made with these resistance genes and how they change the resistance. to try to test the question of, do these genes actually control resistance or not? Next, I delved into the successful defense response, and this is where I used a mix of chemistry, like measuring what kind of chemicals the plants were producing to fight off the aphids, and I also looked at a lot of bioinformatics data to see like how their biological processes were changing in response to aphid attack. I also had what I think is my personal favorite chapter, is how environmental stress impacted the defense response. I like this chapter a lot because there are some very creative experiments done in here, and it is also very relevant to real-world questions of how do we make crops pest-proof when they're under stress from drought, for example. And then a thesis concludes with a general discussion, which is where the writer brings together all of the different strands and has a kind of crossover discussion where we talk about, do certain results contradict each other? Do certain results from the different chapters support each other in different ways? And then often theses conclude with some future directions, and I'm very glad to say that my colleagues at the Turnbull Lab are looking into some of these research questions as we speak. Finally, there's also a big section of references. As you can see, I read a lot of scientific papers in order to write this thing. So there's my thesis. That's a little bit about what goes into a thesis. If you would like to read the thesis for yourself, I'm very happy to announce that it will be publicly available probably later this year via Imperial College London's library services. So if you'd like to read it, just let me know. I'll post the links all over my social media for the rest of you to go check it out. My supervisor and I are also planning to author some publications that will be in scientific journals, and I really hope to be able to do a general audience's overview of my work and perhaps put the presentation online or do it as a live and in-person event. So, once a student has written the thesis and submitted it, which is very exciting, what's next? The final hurdle is to do what is called a viva voce exam here in the British education system. Viva voce is a Latin phrase meaning living voice. So the point of a viva voce exam is for two experts in the student's field of science, in my case plant biology, to come and interview the student to make sure that this is the student's own original work. They want to make sure that the student was able to come up with their own unique scientific hypotheses and to test them through competent experiments. In this modern age of AI, they also want to make sure that the student wrote it and it wasn't written with the help of various AI writing tools. Of course, ones for grammar are fine, but they want to make sure that the student actually wrote this, because it's there can be dodgy stuff going on. But I think there was no doubt that it's all written in my own kind of typical writing style, so they knew that I was the one who wrote it. Also, when the examiners interview a student, they also want to probe how much do they really understand about this field of science? Do they understand how their research fits into the bigger picture of plant biology, for example? Like, what truly new and original contributions has the student added to the field of plant science through their work? Something that I found pleasant and approachable about the Viva Voce is that it really is a conversation with two other people very interested in your research. It's not a defense. I personally had a very positive and I think fair Viva Voce experience. It was based off how I prepared for it. It was all good. Now, in conclusion, you might be wondering what's next for Dr. Katia Hougaard, also known as Katia Plant Scientist. Well, the good news is that although I'm Dr. Katia Hougaard now, I'm still Katia Plant Scientist to you, my fans, and I will be making even more social media content and live and in-person content in the foreseeable future. I plan to keep developing my new career direction in science communication, and I hope that you will join me on that journey as we stay curious about plants together. Thank you so much, and I'll see you next time.